line page, the second packet, begins with the blood vessels. And so if we look at them, remember I reinforced this for you yesterday. What is it that causes the blood to flow from one place to another?
Pressure.
You're so good. Pressure gradient. All right, so when we look at this graph, this real simple graph here, doesn't seem very bright, the arterial system and the venous system at any one given point, if I draw a line across here, the blood volume will be the same here as the blood volume here. But the pressure will be different. So the volume of blood in the aorta and the pulmonary, the aorta leaving the heart will be the same as the volume of the superior and inferior vena cava entering the heart. Otherwise, we start backing up and getting edemas. Okay? So again, even with all the multiple branches of arterioles, if we have 1,000 arterioles, their cross-section, the surface area that the blood has to flow by, is going to be different, but the volume is going to be the same at that particular level. And we'll review that again when we do blood pressure next week. So I want to take you through the basic characteristics of the basic vessels and their functions. So our arteries are involved with pressure reservoir, the veins with volume reservoir, which ones do gas exchange, which ones are involved in diaphoresis, and so on and why. So we'll do that for about 40 minutes, and then we'll take a short break and come back and start following the blood vessels through the body. The anatomy part, learning the names of the blood vessels and the tracing for them. Okay? So our types of blood vessels are arteries, as we leave the heart. And we have two major types of arteries based on their relative distance from the heart. So as we saw with the aortic pressure, the closer the vessel is to the heart, the higher the pressure is, because the pressure decreases as the blood moves farther away from the heart. So our vessels that are closest to the heart are identified as elastic arteries. Also known as conducting arteries. They would include the aorta and some of the major branches close to the aorta, like the common carotids that go up into your neck. Look in the box down there on the floor. And then as we move off of the aorta and into the organs, we lose those elastic sheets, and these are identified as muscular arteries, even though the elastic arteries obviously have muscle. And these are also identified as distributing arteries. Pardon?
This is after the aorta?
Yeah, well, branches off of the aorta are farther branches. Yeah. So, distributing arteries. All right, so if you think of a major freeway, like I-5 as the elastic arteries, and major streets off of that as also elastic arteries, and then you get onto the surface streets, those would be distributing arteries. Okay, so College Oak, Madison, right where it comes onto or off of 80 would be an elastic artery, but where it crosses Sunrise, by that time would be a distributing artery. Okay, so the farther we move away from the heart, the fewer elastic fibers we have, and the more we move into a muscular artery. So this is not just a sudden, you're either this or that. Sometimes there's going to be some slight changes, yes?
Is there a general pressure in correlation to when it's
Probably,
pushing?
but I don't know what it is, because certainly distance is affected by pressure, okay? You don't need to, I'm not going to ask you to identify an artery as an elastic artery or a muscular artery. I just want you to know what the characteristics are. And then we just have our muscular arteries are going to just decrease in size and get smaller and smaller and smaller and smaller. And they're defined by certain diameters. The next structure we're going to look at characteristically is known as an arteriole. And by definition, I'll describe this as having no more than 2 layers of smooth muscle in its wall. arterioles represent the vessel's most important in regulating blood pressure. There are many of them, and they still have enough smooth muscle. Their surface area is greater. So I'll go over that when we get to arterioles a little bit better, why they do that. And then as we lose the smooth muscle from arterioles, we have capillaries. Capillaries have the smallest diameter. They also are the most numerous. So as we're looking at our pathway here, we have the arterial flow pressure in the muscular arteries is going to be greater than pressure in the arterioles, which is going to be greater than pressure in the capillaries. Now as we move from capillaries rejoin to form venules. So is pressure going to be greater in a venule or a capillary? Good. So even though we're rejoining and forming larger vessels, the pressure is still dropping. Because pressure has to drop subsequent in each of these steps in order for blood to return to the heart.
That actually makes sense because it gets larger on that side.
So venules and then veins. And we have various size veins. And then of course we have our large vena cavas. Superior and inferior vena cavas. Now let's look at the characteristic wall component of our blood vessels. And until we get to capillaries, on both sides, arteries or veins have the same three types of layers. Remember we had 3 layers of the heart wall? We have 3 layers of a blood vessel wall. Okay? So the 3 layers are identified as the 3 tunics. The lining is known as the tunica intima. That would be homologous to what layer of the heart wall? Tunica, or other endocardium, okay? And then our smooth muscle layer is known as the tunica media, and that would be equivalent to the myocardium of the heart, which is cardiac muscle. This is going to be smooth muscle. And then we have the tunica externa, which you may also see identified as the tunica adventitia.
And this is for arterioles and venules?
Yes, arteries and veins, including arterioles. Now, typically, just as a real general characteristic, the key difference in thickness between the artery and vein is due to the amount of muscle. And so it's primarily the tunica media that is going to be different when we compare a wall structure for an artery or a wall structure for a vein. And that reflects the pressure resistance that's occurring across those vessels. So if you look at the lumen, let's say that we have the lumen of an artery, and we have the lumen of a vein of nearly identical diameters. The amount of smooth muscle that would be present in the artery versus in the vein would be about like that. Okay, so about 1/3 of the amount of muscle in the artery is required to resist the pressure as we have for the vein. And there'll be a few other, the orientation of the smooth muscle is a little bit different. So here's another view. This is a muscular artery. You can tell because of the presence of a sheet of elastic fibers right at the tunica intima that are associated with that and throwing it into deep folds. You can see that in our tissue slides, the arteries tend to maintain their opening, whereas veins collapse. And again, it has to do with the amount of smooth muscle present. So here's another image of the layers. We have something similar here. This is one of our newer models. Jeff loves to go shopping. And since Jeff took Bio 430, and Jeff is the guy that runs our open labs, took 430 and 431 from me. He's biased towards what I want, so I get to feed him. Hey, I want this model. So this shows the structure from outside to inside of an artery and a vein. And then a vein is cut open here so you can see the valve structures. Okay? On the back, it's showing the effect of muscle contraction on veins. We'll talk about that in a bit. So now let's take these vessels, and I'll diagram on the board and identify the tissue characteristics that we see with the three different components of the vessel wall and why it contributes to that particular function. So the first one we'll look at are the elastic arteries. And the tunica intima of elastic arteries is formed of tissue very similar to the endocardium, though not as thick. We're going to have a simple squamous epithelium. My diagrams are not going to be proportional to size because you wouldn't be able to see the capillary, or I'd have to draw a really huge elastic artery so you would be able to see the capillary, so again, it's not going to be proportional. But we have a lining epithelium, which is simple squamous. And underneath every epithelium is what type of connective tissue? Loose irregular. So that's our standard tunica intima for all of our vessels. All right. Then we add the tunica media. And obviously, since we're talking about an artery, we're going to have a thick amount of smooth muscle. And the smooth muscle cells are oriented so that they are found in their longitudinal orientation around. So we're not cutting, when we cut a blood vessel in cross-section, we're cutting the smooth muscle cell in its longitudinal section. And here, where we're cutting the blood vessel in longitudinal section, the smooth muscle cells are cut in cross-section. All right, so if you were to look at a blood vessel, and we'll see this with the arterial, if you were to look at a blood vessel in longitudinal section, cutting the length of the vessel, you're going to see the smooth muscle cells, small and large circles, because a smooth muscle cell is fusiform in shape. So if we cut here, it's going to create a small circle, and if we cut here, it's going to create a large circle. So when you look at a longitudinal cut of a blood vessel, you're going to see the cells, the smooth muscle cells, cut in cross-section. You look at a cross-section of a blood vessel, you're going to see the cells cut longitudinally. Okay? So rather than draw every cell, I'm just going to do this. We have a thick layer of smooth muscle cells. In an elastic artery, we also have sheets of elastic fibers. Elastic fibers need to be stained with a silver stain to be seen, so they are black. And you can see this staining on your lecture notes. There's a photograph, even in black and white, you can see the dark lines of the stained elastic fibers. No, go back. You want to go back. Those are PowerPoint slides, lecture notes. So page 74 of your lecture notes. So the purpose of this elastic, since these vessels are under such high pressure so close to the heart, it's not so much for the expansion as to help them recoil as that blood is helping it to move on. So elastic has a return to normal length factor. All right, yes, it can stretch, and we often tend to think of the elastic stretching capability, and we forget about its recoil. And it's that recoil component that is important. So we look at a elastic band, a rubber band, and you think of that as the elastic fibers around a blood vessel wall. And as the heart pumps blood into the aorta, it stretches it, and then as that blood it comes back, that elastic recoil helps to compress the blood and continue to move it through. Okay, so that's its importance here. All right, now let's look at muscular arteries. So I'm going to leave up most of this. I'm going to erase the elastic fiber sheets. Oh, tunica, let me put the tunica externa here first. Tunica externa in this large vessel is going to be dense, irregular connective tissue, forming a thin capsule that helps to hold everything in place and anchor the blood vessel in the muscle or along the chest wall, wherever it is.
Did you say helps to anchor blood vessels?
To its surrounding tissue. So now we're looking at muscular arteries, which are most of the arteries in the body, are going to be muscular arteries. And back to this previous image, which was of a muscular artery. Notice how ruffled the lining of the muscular artery appears to be. And if you have these slides on PowerPoint, you can see there's a dark purple line just deep to the epithelium. So we make those changes between elastic and conducting arteries. So this is thrown into fold because of a sheet of elastic fibers that are found just deep to the tunica. It's actually a component of the tunica intima. So I'm going to add this here, the internal elastic lamina. And we still have a loose, irregular connective tissue. And then we have our tunica media without the elastic fiber sheets. All right, so in the muscular artery, the tunica media is not going to have there might be an occasional elastic fiber, but not the prominent sheets. So this would be the tunica media. Here you can see an occasional elastic stain in there. I can't even make it close, but not whole sheets of them. And then we would have the tunica externa. The dark blue are collagen fibers. So lots of the sheets of elastic fiber, addition of the internal elastic lamina. I didn't mention it to my lecture this morning, but there's also an external elastic lamina between the tunica media and the tunica externa. You don't need to worry about that, but just for accuracy's sake, it's there. Different places, yeah, different amounts. All right, so these are going to get smaller and smaller and smaller and smaller. It's described as large arteries or small arteries. Once the muscle wall is thin enough that only 1 to 2 layers of smooth muscle cells are present, that determines that it's an arteriole, right? That's the classification I use based on a histology class that I taught. You might, if you Google this or Bing search or whatever internet search, and you put in arteriole, you will find images labeled, or blood vessels labeled arterioles that are 3 or 4 muscle cells thick. If I show that to you in a lab quiz or an exam, it will be an artery. It has to have only 1 or 2 smooth muscle cells in the tunica media to be able to fit the classification of arteriole that I use. All right. So let's change this now to arteriole. I spelled it this way this morning, and nobody corrected me. Until I looked at it and went, wait a minute. The diagram of the arteriole in your lecture notes is one that's cut in longitudinal section. So you will see cross sections through the muscle cells rather than longitudinal sections. It will have a regular tunica intima. So I'm going to draw this a little larger than it would be in proportion just so it's visible. Simple squamous epithelial cells. We've lost that corrugated look because we don't have the internal elastic lamina. We still have loose, irregular connective tissue. However, the tunica media will have just 1, maybe 2 layers of smooth muscle. Okay? I'll write that. Because there are so many arterioles in comparison to even small arteries, this makes the arteriole the site of the autonomic control of blood pressure. So next week when we talk about blood pressure, if we decrease the diameter of an arteriole in half, we increase the resistance by 4. So we'll talk about further. In other words, I don't know how many grew up with the magic school bus, but my kids did. There was a show on resistance where they had the kids playing baseball on an ice skating rink. And of course, they're able to slide faster than they could run. So if I were to take these scissors, slide them across the table, it's peripheral resistance between the scissors and the surface of the table that slows it down and eventually causes it to stop. All right. So if I have a blood vessel of this diameter, and I have the same amount of, that's our artery, and I have the same amount of blood flowing in, 10, 9, 8 arterioles. The volume is the same, but there's an increased amount of surface area. So there's going to be more resistance. Because I still have some smooth muscle, I can decrease this diameter much more than I can decrease that diameter. So the arteriole becomes a vessel of regulation of blood pressure through the sympathetic nervous system, as we'll see next week, causing constriction or dilation. There's always a slight tone. So when blood pressure drops, the vessels will dilate. When blood pressure when blood pressure rises, the blood vessels constrict. So if you're scared, your face doesn't get red, it gets pale. Okay? So at the same time, we're constricting blood supply to the skin via the arterioles. We're dilating arterioles in the skeletal muscle to increase blood flow to the skeletal muscle. So we'll look at that effect on blood pressure next week. But I want you to recognize that that is the major function of the arteriole. So arteries in general are function as our pressure reservoir. We need to increase or decrease blood pressure. That's where we change parameters. All right, now let's remove even the smooth muscle. So we'll have no smooth muscle, all right? And we'll have no tunica externa. We don't have any smooth muscle. And the vessel we're left with is identified as a capillary. So it's formed only of simple squamous epithelia. It may take 1, 2, or 3 simple squamous epithelia to form that wall. Attaching the epithelia to each other.
Is it capillary or artery?
Capillary.
Capillary. Attaching the epithelia to each other are tight junctions. And as you recall from the brain, the more tight junctions we have, the smaller the molecules are that can pass in or out of the vessel. And we have the blood-brain barrier. Antibiotics don't cross the blood-brain barrier. Dopamine doesn't cross the blood-brain barrier. So we can have leaky or tight capillaries. And in your lecture notes, as well as on the PowerPoint slides, there are 3 types of capillaries that are illustrated. So this would be a typical capillary over here. Endothelial cell is our name for the epithelium lining blood vessels. Here's one cell, here's another, and at their junctions, they're joined by tight junctions. Every epithelium, remember, has a basement membrane. It's one of the characteristics of epithelia. And so if there's a full basement membrane present, it's identified as a continuous capillary. Full or complete basement membrane. Maybe I'll make that to complete. If the basement membrane is incomplete, if it's patchy, like you see on the lower diagram here, then it's called a discontinuous capillary. And we usually see this in sites where capillaries are present that large molecules need to pass across the membrane. So in this one, you can see there's large spaces in the membrane and between the capillary cells. We find this type of a capillary in the bone marrow or in the liver, where red blood cells move in and out, or white blood cells that are newly formed in the bone marrow. And so a very large capillary like that is also known as a sinusoid. So capillaries are typically slightly larger than a red blood cell. Anybody remember the diameter of a red blood cell? About 7 microns. So this is going to be between 7 and 9 microns in diameter. A typical capillary, whereas a sinusoid may be 10 to 12. So in a typical regular capillary, having such a small diameter causes the blood flow to slow down, and the red blood cells have to travel one by one. Kind of stack up. And that slows it down and gives more time for gas exchange. Right. So the oxygen that's moving out of the air sacs in the lungs has more time to enter into the red blood cell, and more of it. And if it's in the tissues, there's more time for the oxygen to leave, and shorter distance.
Contact, surface contact.
Yeah. Okay, so now up here, you'll notice that it has a continuous capillary, a basement membrane, but the cells have small channels in them. And those are known as fenestrations, fenestra, or pores. So this is specifically an example of a capillary in the kidney. We'll talk about it where it functions in filtration. So both the basement membrane affecting charges and the size of those pores determine what leaves the blood. So in other words, in the kidney, ions, individual amino acids, can pass out of the capillary, but proteins and red blood cells are too large. All right, so that this is affecting transport across the membrane. Now, let's take our capillaries and